start by some motivational slides that are intended to be provocative also. Originally I was a physicist by training, and my first entry into the field of biology was actually through theory of evolution and modeling of evolutionary dynamics. And there is basically now almost 100 years of theory called mathematical population genetics. And so I started to work in this area, and over time I started to get sort of a bit disturbed by the very, very different relationship that you find between theory and experimental data when we're talking about the theory of evolution and when we're talking about other physical theories. In particular, it's still exceedingly rare for evolutionary theory to make concrete quantitative predictions about things that you expect to see in nature. All right? Such things happen, but it's actually very, very rare. And I'm starting to wonder, okay, why is this? Why is there this very, very sort of weak connection between theory and experiment in evolution? And at some point, I was reading in the part 1 of the lectures on physics of Richard Feynman, and he is explaining to the undergrads how physics works in his sort of Feynman-esque simple way. He says, well, you know, what we do in physics is the following. First, we observe phenomena that interest us. Then the first thing we do is we formalize these phenomena by finding quantities that we can measure in a reproducible way. So that's step 1. You find things that you can measure about the things you're interested in, that you can put numbers on, and that you can do in a reproducible way. That's step 1. Then step 2 is you just start to look phenomenologically for relationships between such quantities, okay? So go to Galileo, you drop a ball, the distance that the ball has traveled goes like the square of time, independent of how, you know, you hold or if you drop it from the Tower of Pisa, this is a case. And once you've now collected a large number of such quantitative relationships that you've just observed, the glory of physics is when you find a theory that in one full sweep explains many, many of such quantitative relationships. All right? So this is the way in which basically all physical theories in the past have been developed. And if you now compare this with the situation in evolution and math population genetics, you find something that's very, very different, right? So there is no collection of phenomenological laws about quantities that you can measure about evolution that the theory is supposed to explain. It's often not even clear what measurable quantities about organisms in the wild you should look for to make such laws. What has happened is that basically there was a qualitative framework developed by this gentleman, Charles Darwin, and people just started making up mathematical toy models and say, let's imagine there's one gene and there's 2 states of this gene, and one state has this fitness and another state has that fitness, and it mutates at this rate. What do I expect to happen? And what I think is the most worrisome is that the key quantities in these mathematical population genetics models, like fitness or effective population size, are essentially impossible to measure. All right? And there is an agreement in the field on this, that nobody knows how to measure these quantities. So now, I'm being intentionally provocative because I've worked myself a lot in this area, but I'm thinking that if you're developing your theories and the central variables in your theories are unmeasurable, you're putting yourself up for trouble, okay? This is really a problem. Now, so what I propose...
Erik, since
yes.
This is a provocative slide. Yes. Probably this is true for all of biology.
No, I don't think quite as much.
Well, why only evolution? I mean, development or cell or neuroscience?
I think in a lot of biology, there has been much less pretension to have a complex mathematical framework. right? So the early molecular biologists, they just wanted to understand these phenomena that they saw in biological systems in terms of the underlying physics and chemistry. And they weren't really sort of worrying about deep mathematical models. And in fact, most molecular biologists, until recently, when there were very, very large data sets that they don't know how to handle without mathematics, they were very skeptical of any kind of theory, right? They would tell you jokes about when the genetic code was discovered, there were theorists that were predicting how it was going to work. Why are there, you know, how the 64 codons are going to map to the 20 amino acids, right? And they were all wrong, right? So most molecular biologists are quite skeptical of how much theory can help at all in these systems. And evolution is really one of the areas where there has been the largest, longest tradition of theory development.
But the first two things about reproducible measurements and
yes.
Actually building theories for measurable quantities.
Yes.
That probably is true for other areas too. Anyway, I mean, this is, I don't expect a definite.
No. So, but I do think, I mean, but good molecular biologists take a lot of care in making reproducible measurements and setting things up using the right controls. And in many cases, this is why experiments are set up to give yes-no answers, because they know they cannot really get to sort of like a quantitative measurement of how much does this vary as I perturb this. And so they really say, does it change or not change? And then they show that they can reproducibly measure and so on. So I do think that good people do this, right?
Erik, just a quick question about fitness. Does your statement also apply to very specific, simple cases where you grow 2 strains of bacteria and measure their relative, population size in the subsequent generations? Even this applies to that situation also?
Yes.
And why is that not a good measure of fitness? Just the relative.
Okay, so I don't want to spend the entire 1.5 hours on this slide, and we easily could. But so, for example, so in the experiments of somebody like Richard Lenski, he really takes great care to take a simple lab strain out of which, active phages, transposable elements, and so on, that all very well behaved, not all this, because you put E. coli together, they start fighting, right? I mean, they interact in ways. So to have a simple situation and so on. And there's some recent work actually that came out that shows, even in that situation, it happens generically that you get ecological interactions between subpopulations within your little shaken tube where there is only glucose, and you have coexistence of strains and so on. And when they do these kind of measurements, they can get non-transitive results. A does better than B, B does better than C, but C does better than A, and so on. So it's very, very hard to find situations where you can really measure a number and say, that number is fitness, and it really is fitness like the way it enters in my theory. All right, so the only thing that I sort of want to do is to say, I think that it's worthwhile to try and focus on this part, okay? What are measurable quantities about evolution in the wild that we can look at? And can we find phenomenological laws relating these quantities? Okay, let's just try to first, get the Galileo laws and the Kepler laws and these kind of things for evolution before we imagine what kind of theory, right? And this is also for like a lot of physicists coming into this field, like myself, they all imagine that they work on this part, on step 4, and that they're going to come up with some grand theory that explains everything, right? But we haven't really done this yet, I would argue. All right, so the second thing I want to point out, there is really an opportunity to study this, because since sort of the mid-90s, we have had access to complete genomes. And one of the beautiful things is that essentially the entire result of the evolutionary process is stored in the DNA sequence. right? So it's not just a part of the information that evolution has acted on, but it's essentially, to a good approximation, everything that evolution has acted on. And the number of available genomes has been increasing exponentially with time, even faster than this Moore's law for computers. And so by now, you have many tens of thousands of complete bacterial genomes that are available. So there is a lot of data available to just look at what is in that data. Are there any phenomenological laws there? Now, one challenge is that if you get your genome, this is what you get, right? So you just get a very, very, very long strings of A's, C's, G's, and T's, right? And so you have to make something out of this amorphous information to really start asking questions about what kind of observable quantities should I look at. And so here I want to make one other mention, is that especially under my physics colleagues, these sort of bioinformatics methods for analyzing sequence data are often thought of as just really sort of elevated bookkeeping and not, you know, not very, very deep science. But I do think that they have made enormous accomplishments over the last decades in going from this to having quite some understanding of what occurs in a genome. So nowadays, if you sequence a new bacterial genome, you have easily usable bioinformatic tools that will tell you where all the genes are in the genome, translate them into amino acid sequences for you, find out what other genes there are in other genomes that are related to your genes in your genome, and find out the evolutionary relationships between the genes in your genome and the genes of other sequenced genome. Find out, at least to a rough approximation, what kind of function your protein has. So they will tell you if your protein sits in the membrane, whether it's a transporter, whether this is a metabolic enzyme, maybe it's a transcription factor. These kind of rough functional annotations we can now automatically get for the very large majority of proteins. And we often even know what parts of a protein, which domains in the proteins are responsible for which parts of this function. And in many cases, we can even get an idea of the three-dimensional structure of the protein, and C has been talking about these things. And so ultimately, all this is based on pairwise and multiple sequence alignment and putting gene sequences into families and modeling the families and so on and so on. So I just wanted to sort of, you know, pay some respect to these methods, because I think it's actually quite amazing how many things we can really extract from the sequences nowadays using these tools. Okay, but I'm not going to talk about how you do this. I'm just going to talk about what you use from the results. So there are these databases, right? So for example, here's the NCBI database of microbial genomes. So you can go there, you can get these thousands of genomes that have been sequenced, that you can download them with the annotations of what genes there are, what the functional annotations are of these genes and so on. So there's all this information ready for you to look at and to start looking for phenomenological laws, right, in genomes. So I will tell you about some things that people have observed when they started looking at this. So one of the simplest questions you can ask is, how many genes are there as a function of how big the genome is? Okay, so, and this is just from the database. So this is 10 to the 4 base pairs, 10 to the 8 base pairs is the total genome size, is the number of genes. Here is 10, here is 1,000, here is 100,000. And you see that in viruses and in prokaryotes, so bacteria and archaea, it's to a very good approximation linear. So the size of the genome is just a linear function. So the number of genes in the genome is just a linear function of the total size of the genome. There's a fixed length of genome per gene, which is about 1 kilobase. It's also nice. So you have to divide by 1,000. If you get the genome length, that's the number of genes. All right? In eukaryotes, you see that there is some bending over going on, right? So once you go to big eukaryotic genomes, the number of genes is going up actually much slower than the genome size. So there is a lot of stuff that is outside of genes, which you don't see in bacteria and viruses. All right, so now you can also start taking genomes and comparing them with each other. So here's just one example where I randomly took a genome, Carboxylothermus hydrogenoformans, one of the genomes in the genome, took the origin of replication of this gene, so these are the genes near the origin of replication in this genome, and compared it with E. coli. That's a E. coli as well. Lab strain that many, many people study. Yes. Okay, so most bacterial genomes are circular, and there is a single spot where a complex will assemble that will start replicating the genome. And so the genome is replicated from this one position, and basically machines are running down this circular genome in two ends and making a copy of this genome. And there's a single place where this replication starts, and it's called the origin of replication. So here are the genes around the origin of replication in this genome, and here I found a part in the E. coli genomes where you find that clearly similar genes are occurring in the E. coli genome in also a very similar arrangement. So the arrangement are not identical, right? There are some things here that are not matched in between, but you see that many of these genes that are sitting near this origin of replication can be identified with each other. And it's quite remarkable, right? Because this genome is one of the most distal genomes to E. coli. It's very, very distal. So there's more than 1 billion years of evolution that are separating these two genomes. And still, this architecture of the genes near the origin of replication has been maintained. All right. So now what does it mean, right, that I put these sort of arrows between these two genes, that I think these genes are similar? So what it means is that we think of these genes as orthologous genes. So what we imagine is that in this ancient ancestor genome, that is the ancestor of both the C. hydrogenophormans and E. coli, these genes were already present, copies of these genes were already present, and they were probably performing very similar function. And as these genomes have evolved independently of each other, these genes have been maintained in a similar organization. All right? So we call these orthologous gene pairs. They came from an ancestor gene that existed in the ancestor of these two genomes, and in most cases has retained much of its structure and function over this evolution. All right, so now once you have orthologous pairs, I can start comparing how different do these two genes look, right? So I can align those orthologous pairs with each other, the gene sequences, and just ask how many positions have changed and how many positions have stayed the same. So you can do this for all orthologous pairs, and you can make a histogram of the rate of evolution of different genes. All right, so this figure I got from Alan Drummond, and where he did this for two species of yeast. So Saccharomyces cerevisiae and Saccharomyces baianus, they have a common ancestor about 20 million years ago. And so what he did is he looked for orthologous pairs in these two yeasts, and for each orthologous pair, estimated the rate at which positions in these genes have been changing during evolution. And he separated that in a rate at synonymous positions. So these are changes that do not change the amino acid sequence. So Harmeet talked about this also a couple days ago. And non-synonymous changes, these are changes that change the amino acid, and made histograms of their distributions. And in these dotted lines here, you see what this histogram would be if all proteins were evolving at the same rate. All right, so if all positional proteins had a constant rate of changing, then you would get the Poisson fluctuations in the number of changes that you see between a pair. But you see much wider distribution of changes, right? And not only in the synonymous one, but especially in the non-synonymous ones, there are almost 3 orders of magnitude variation in the rate at which the protein is evolving. So some proteins are evolving very fast relative to other proteins that are evolving very, very slow. And yes. Yes, correctly, correct. And something that was then pointed out by Eugene Koonin's group is that if you take these distributions from very, very different pairs of organisms, so you take a human and a mouse, or you take two bacteria, or you take two plants, or you take very, very different organisms, and you rescale them such that the mean of this is 1, right? So this is on a log scale, notice, right? So this is the log rate of substitution. So you basically shift the distribution such that the mean is at 1. Then for pairs of genomes, you get really quite similar looking distributions, even though they're taken from very, very, very different organisms. Yes?
It's only scaling the mean, not the width. Is that right?
Okay, so this is on a log scale. Right, so this is 0.01. So this is, the substitution rate is on a log scale, and so you're basically just shifting the distribution over. You see that the variance of these distributions is actually quite similar. All right? And so it seems that there is a sort of almost like a universal, roughly log normal distribution of evolutionary rates that vary by about 2-3 orders of magnitude across genes in the genome. So that, that's just an observation, right? So that's one of these laws. So why is this? Why are some genes evolving very slow and other genes much, much, much faster? So of course, there have been a lot of people, once this was clear, they have been studying why is this? Why are some genes fast and some genes slow? And to date, I think the best correlate that people have found that determines whether a gene is fast or slow is its expression level. All right, so this is from a paper of Alan Drummond and Klaus Wilke, where they show for yeast and E. coli the dN and ds, so these rates of substitution at synonymous and non-synonymous size, as a function of the mRNA expression level of the genes. And so even though this is very, very noisy, right? So if you look at it, you said that's not a great correlation. That's the best correlation people have been able to find of any variable that a gene has and its evolutionary rate. So you see that high expressed genes tend to evolve slower than low expressed genes. Okay. Similarly, they looked at the codon bias of the gene, and they find that genes that are high expressed have much more codon bias than genes that are low expressed. That is to say, for every amino acid, there tend to be multiple codons that you can use, and one of these codons is the favorite codon. It's the one that is used most often, and it typically corresponds to the codon that has the highest concentration of tRNAs. And you find that the bias that the genes have of using the favorable codon is higher in high-expressed genes than in low-expressed genes. All right, so why might this be? Why is there this correlation between expression level and rate of evolution? Yeah.
Yeah, there's still some massive variation about the there's massive variation around those straight line fits. I mean, how much of that comes because of error in just measuring the mRNA levels, and how much of it is because of deviations from the fact that misfolding isn't the only evolutionary constraint?
I think mainly the latter. There's not much error in the measurement here. Of course, you can say, well, mRNA level in what environment? They take one, and maybe, you have to worry about that kind of stuff too, right? But I think it's just that there are other things that determine this that we don't understand yet. That's my feeling. So the explanation that they provided highlighted the fact that compared to the DNA polymerase that replicates the DNA and the RNA polymerase that transcribes the DNA into mRNA, the ribosomes that translate mRNA into proteins are very, very error-prone. Okay? So they make errors of magnitude higher rate than these other polymerases. So it's somewhere between 1 in 1,000 and 1 in 100 even of amino acids that are added to a protein chain are wrong. Now, you can imagine that if you make a protein that is a few hundred amino acids long, and you put 1 or 2 amino acids in there that are the wrong amino acids, this may cause your protein to no longer fold. And when your protein doesn't fold anymore, it's not functional, so you have to make a new one. And second, it may even be toxic, such a misfolded protein. You have to make sure to get rid of it. So these misfolded proteins incur a non-trivial cost. And the genomes, right, the organisms can evolve their genomes to try and reduce this rate of misfolding. So for a given gene, you may reduce the rate of misfolding either by making sure to use codons that are unlikely to be mistranslated. So if you use the favorite codon with the highest number of tRNAs, it's less likely to introduce an error than if you use a non-favorite codon. And second, to maybe use a combination of amino acids that folds extra stably, so that even if you put one or two wrong amino acids, your protein will still fold correctly. All right? So this is what you can do as a gene to try and lower this rate of misfolding. But now notice, if you lower your rate of misfolding from, let's say, 10% to 5%, this is a lot more misfolded proteins that you avoid if you have a high expressed gene than a low expressed gene. All right? And so these high expressed genes are effectively under a much stronger selective pressure to adapt to lower their rate of misfolding than low expressed genes. And of course, because there are many fewer sequences that are highly adapted to avoid misfolding, this will lead to a lower rate of evolution, because there are now less changes you can make to your gene that keep your gene equally stable. All right? So that's, I think, so far, the most plausible explanation of why there is this systematic variation in evolutionary rates and why gene expression level is actually a major determinant of the evolutionary rate. Right? So this is just one example. Okay, yes?
So which kind of codons are more likely to be mistranslated? How do you determine which codon is more likely?
So actually, this is not really my area of research, so I don't really know the details of the chemistry of the codon recognition. But I think a first order approximation is the different codons that correspond to one amino acid have different, just different concentrations in the cells. And so if you use the one that has the highest concentration, then the waiting time that you have before the right codon diffuses into your ribosome is less than if you use a codon with a very rare tRNA. And so that's one way, for example, in which you can lower this. All right. Yes. Is it about the teleological sort of arguing when you talk about evolution that bothers you?
No, so it all comes down to the fact that there is a higher proportion of a certain codon that gives rise to the higher translation of that specific gene, right?
It's not necessarily about the rate of translation, right? So the ribosome is just sitting there, and it has one codon of the gene sitting there, and it's waiting for tRNAs to diffuse into this pocket where the reaction can take place, and it has to decide when the right type of tRNA has diffused in. And this is done by the energy of interaction between the codon and the anticodon. But of course, you will have many times the wrong one come in first, and you have to sort of say, no, this is the wrong one, and wait until the right one comes, and then you hydrolyze the ATP and do the reaction, right? And of course, now, sometimes you make an error, and you put the wrong one, right? Because it's all finite energy differences that we're talking about, so this will happen at some rate. But of course, if the right codon is very abundant in the cell, then it's going to likely show up much earlier. You don't have to wait and reject many before it comes. I don't want to spend the whole lecture on this. This is quite a detail now, but the thing. So, yeah. Okay, so if I lower the rate of misfolding from 10% to 5%, say. But I have a gene that has 1,000 mRNAs in the cell, then that corresponds to 5% of 1,000, right? 50 events. If I have only 10 mRNAs per cell, then that only corresponds to half an event. So it pays much more to optimize you lower the misfolding rate for high expressed genes than for low expressed genes. All right, so here's another phenomenological law or observation. So this is actually my first paper in biology as a PhD student. So I did this together with a friend, Martijn Huinen, and he noticed that if you take the few at that point in time, these were all the genomes that were available, all of them, right? And you go and look in this genome, and for each gene, you ask how many genes are there in the same genome that are similar to my gene, right? So you estimate what is the size of the family that this gene belongs to, and you look at the histogram of the gene family size distributions. So how many families are there of size 1, 2, 3? So families of size 1 is a gene which there is no similar gene in the genome. Family size 2, there's 2 genes that are similar, and so on. These distributions are roughly power laws. And another observation, this is newer and this is not published, but one of my students made this at some point. If you measure the slope of this power law, this exponent beta here, as a function of the size of the genome, the total number of genes in the genome, there is a clear relationship. All right, so these power laws are steeper for small genomes than they are for long genomes in some characteristic way. And as far as I know, nobody has a model for this function. I don't have a model for this function. We tried a bit, but it's not obvious to us why there is this dependence of this exponent on genomes. But so this is like a Kepler law, right? Okay, so there is this kind of dependence, it's observed, and it's something that you might want to find an explanation for. So the other thing that I want to quickly mention is that often when you now want to go and look at the functions of proteins, right? So this is something that I wanted to do. I didn't want to just count genes and similar genes, but I wanted to also look at the functions of these genes. Typically what people use is they look at that at the level of protein domains. And protein domains are a nice sort of level to look at instead of whole genes, because genes are typically combinations of protein domains. And protein domains are structural units in the sense that if you just take this piece of the amino acid sequence, it will fold by itself. They're functional units that they typically implement some function like this domain binds DNA, or it's a kinase domain, or it's a transporter. And third, they're atomic in the sense that you cannot split the domain in half and still have something functional. Right? So you can have multiple domains and make a gene that has multiple protein domains that have different functions that they combine in one gene, but you can't take apart a protein domain. All right, and so by now there are these very large bioinformatic resources that have made mathematical models. These are profile hidden Markov models for a very large number of such protein domain families. And there are tools like this hammer from Sean Eddy that allow you to, when you have a genome, basically run these models over all your genes and predict how many copies there are in your genome of each of these protein domain types. All right, so this is one of the things that I used to start counting across different genomes how many genes are there involved in different biological functions. Okay. All right, so here I'll show you some of the results I found. So in this picture, on the x-axis is the total number of domains in the genome for which there is some functional annotation. Each dot is a genome, so here is basically the total number of genes in the genome, and here is the total number of genes, or more accurately domains, that are involved in a particular function. So in blue, you see how many metabolic process protein domains there are in the genome. Here, how many domains involved in translation, and here in red, how many involved in transcription regulation. And so what you should notice is that these axes are both on logarithmic scale, and that these scatters seem to look like roughly straight lines. All right, so what that says is the logarithm of the number of genes in a category is a linear function of the logarithm of the total number of genes in the genome, or the number of genes in a functional category scales as a power law in the total number of genes in the genome. And different functional categories have different slopes. Just 1 minute, I'll get to you. So for example, for the metabolic process, the slope is close to 1, it's slightly below 1, but it roughly says every time I double my genome size, I double the number of metabolic enzymes. So maybe that's sort of what you expect. But here for transcription factors, the slope is close to 2. All right, so that says that the number of transcription factors scales quadratically with the number of genes in the genome. Every time I double the number of genes in the genome, I get 4 times as many transcription factors. It's like administrators in an organization. Right? Every time the whole organization doubles, you get 4 times as many administrators. Whereas for translation, right, it's almost constant. All right, yes.
How do you change? These genomes are from this different species?
All the genomes, at that time when we did this, 2008, this picture is from, these were all the prokaryotic genomes we could get our hands on. You just download them from the database. So some of them are closely related. Some of them are very far apart. And it's just that somebody has decided to sequence that thing. So it might be a biased collection of genomes, right? There might be more pathogens in there than other things and so on. But it doesn't really matter, right? I mean, they still fall on these lines. Yeah.
Basic question. So what is a domain basically? It would be number of collection of genes, a domain?
No. A domain is a part of the protein that if you cut it out, still folds into a defined shape by itself, and performs some function like bind DNA or catalyze a metabolic, an enzymatic reaction, or puts a phosphate on another protein, like the kinase domain. So a domain is just a piece of a protein that has a well-defined shape and function.
So when you say domains in genome, you mean the genes that code for that protein?
No, I'm saying that, okay, so if you take a dot here, then it says, here's a genome in which I have 2,000 domains for which I have any functional annotation. And there are about 200 of these that are actually transcription factor domains. These are domains that code for a DNA, that bind DNA and are involved in transcription regulation. That's what this means. Okay, so if I go to this small genome where I have only 500 annotated domains, I find only 20 transcription factors. And I go to this very large one here with, 4,000 annotated domains, I find 500-600 transcription factors. All right, so in bacteria, if you go to the smallest bacterium with about 500 genes, it has about 1 transcription factor. If you go to the biggest one with about 12,000 genes, it has something like 1,300 transcription factors, which you should compare with, like, human has like maybe 1,700 transcription factors, and a fly has only 600 something. Okay, so some bacteria have more than double the number of transcription factors that a fly has. All right. No. These are all occurrences of the domain in the genome. Of a domain family. Of domains families that belong to, that are transcription factors. Yes? All right. Now, you may think maybe I just picked 3 categories where it comes out looking nice, but that's not true. So for almost any high-level functional category, that is any category where most genomes have at least one gene in that family, all right, you find that something like 90%, 85, 90% of the variance in this scatter can be explained by a linear fit. All right, so it's the rule rather than the exception that the number of genes in a functional category grows as a power law in the total number of genes in the genome. And if you look at what the exponents are of these power laws, you find that sort of metabolic processes, they have slopes that are close to 1. They roughly scale linear with the size of the genome. Processes that are very basic, like translation, DNA replication, DNA repair, these functions that everybody has to do, every cell, the number of genes in those categories grows slowly as a function of genome size. Whereas on the other end of the scale, the fastest growing categories are transcription factors and signal transducing genes. So genes involved in regulation are growing with genome size by far the fastest. That's the sort of pattern that we see. All right. So what does this now say for the evolution of gene content over time? And here I will try to use the blackboard a bit. Sanjay asked me to use the blackboard more often. So I basically just wanted to work out the most basic mathematical consequences of the fact that these that you observe these power laws. All right, so what I'm going to look at is I'm going to take every genome that I see today and think about its evolutionary history. Right, so there is some sort of tree that relates all the genomes that I have in some phylogenetic tree going back to the common ancestor of all my genomes. And I can sort of trace back the history in this tree of every one of my genome. And I'm going to think about two quantities. So for a particular, I'm going to look at the number of genes in the ancestor of this particular genome that lived at time t. All right, so this is to say if I go to the parent and the grandparent and the great-grandparent, and I go back to time t, how many genes were in the genome of that ancestor? And for a particular category of interest, how many genes were there in this category of interest? And then I'm going to look at the logarithms of these quantities. So I'm going to define x of g and t is the logarithm of this n of g and t. And xc of g and t is the logarithm of nc of g and t. All right, so now what is the observation? The observation is that xc of g and t is beta c, some constant, plus alpha c, this is the scaling exponent, times the x of g and t, where t is now, right? If I take a genome now, and this is true for all g. Okay, so it says there is a single function, a single linear function, that relates the logarithm of the number of genes in this category to the logarithm of the total number of genes. Okay. So now I'm going to say, well, this x of my genome t today is simply the integral from 0, which is the common ancestor, so I put the time of the common ancestor at 0, to t today of t now. Today is now. Now it's today. All right. I forgot. It's xg at time 0 at the common ancestor plus the integral from 0 to t now of dx g comma t. I mean, I'm not doing anything. I just do math. Okay? I'm not making any assumptions. But because I know this is log of n, right, I can replace this with log of n. And the d of log of n is just the d of n over n. And I can do the same for a particular category. I mean, it's just writing the same formula for one particular category, but now there are just these subscripts C hanging on all variables. Now, the fact that this has to hold for all genomes means, well, okay, so it means two things. First of all, you can see that the beta C is related to the initial conditions. All right? In order for the beta C to come out right, I have to say that all genomes started from the same initial conditions. But that's easy, because we know they all started from the same common ancestor, so they did all start from the same initial conditions. So that part is taken care of. But in order for this part to also be the same for all genomes, what I need to have is that this integral is equal, so dn over n from 0 to t to now is times alpha c is equal to the integral of dnc over c. All right, so basically what I'm saying, if I go over the evolution, and this has to be true for all g. Right, so if this depends on g and t, this has to be equal for all g. So what it says is, as I go along one of these evolutionary histories in my tree, and at each little time interval, I look at the change in the number of genes in the genome divided by the total number of genes, and I average that or integrate that over the entire history. Or I do it for one category. So if I look at the change in the number of transcription factors divided by the total number of transcription factors and integrate that over the entire history, then these two quantities are proportional to each other. All right? With the proportionality constant, which is my scaling exponent. And that has to be true for all genomes. All right? So this sort of says that when you average over enough evolutionary time, the rate at which you're adding or removing transcription factors relative to the total number that you have, so the rate at which you're growing the number of transcription factors that you have, has to be, on average, twice as big as the average rate of growth of all genes in the genome. Okay? So that's the interpretation of these scaling laws. And now you may ask, okay, so what if I now make one additional assumption? And the additional assumption I want to make, one second, is a time invariance postulate. So I'm going to assume there is nothing special about today. If I were to have gone back 200 million years and I'd sampled genomes 200 million years ago, I would have gotten the same scaling laws. If I make this assumption, then you can say that instead of these integrals have to be true, this has to hold also over small periods of time. It has to be invariant in time. And when it's invariant in time, it's basically says the change in the number of genes in this category divided by the total number of genes in this category is equal to the change in the total number of genes divided by the total number of genes times the scaling exponent. All right, and you can rewrite that as saying the change in the number of genes in category C per change in the genome is in fact this fraction of all genes in the genome that belong to this category times this scaling exponent. Right, so if you think of a sort of a null model, the null model would be whenever I add a gene to the genome, it's a copy of a randomly selected gene in the genome. Let's think of this sort of genes randomly duplicate or delete. So whenever I add a gene, I randomly pick one. Well, then what I would get is this equation, but without the alpha C. Then the probability that it will be a gene in this category is just equal to the fraction of the genome. So what the scaling law says, it says no. Some genes that are in a category with low alpha C, they're systematically not taken to be duplicated or deleted. Right? They tend to not be duplicated or deleted. And some other ones, like transcription factors, they're twice as likely to be picked for duplication or deletion. And so this is sort of what these scaling laws, I'd like to interpret what the meaning is of these scaling laws. Now, if that's true, I should be able to test this by looking at close pairs of genomes. I take pairs of genomes that had a common ancestor not very long ago, and I try to estimate in this pair of genomes what were the total number of additions and deletions that have taken place. By just comparing these genomes, and their genomes are almost identical, I say, oh, there were two transcription factors added and one metabolic enzyme removed, and so on. So I can look at what were the total number of changes and what fraction of these changes corresponded to genes in each functional category. And the definition is then, if you take the fraction of the genome that corresponds to genes in category C, and you take the fraction of all changes that belong to genes in category C, the ratios of these two fractions should be equal to this scaling exponent. I call this the evolutionary potential. So some categories have a lot of evolutionary potential. They're often added or removed. Some have low evolutionary potential. They tend to not be added or removed. All right, so the prediction is that this evolutionary potential that I here define by this fraction of observed changes that involve genes in category C in this close pair, divided by the fraction of genes in the genome that belong to category C, that evolutionary potential should be equal to the scaling exponent. So the next slide shows you that, well...
Erik,
yeah.
Sorry, I didn't get that. Can you go back to the last slide? How do you know that that is independent of time, the delta n?
Okay, so that's an assumption that I now want to explore. I say, if I assume that this is time invariant, then it also has to hold over short periods of time. So let's look at guys that just differ by short periods of time and see if it holds. All right, so this is what my student Nacho Molina did. And what he found is that indeed there is a good correlation between the exponents that we fit to a category and this evolutionary potential. I think it's important to realize is that this number is gotten by comparing all genomes and seeing how the number of genes in this category changes as a function of the total genome size. Whereas this quantity uses completely different data. You take pairs of genes that are closely related, look at the small number of changes between them, and you look at the fraction of these changes that involve transcription factors. So it's a very different kind of data, right? And the fact that you find this good correlation says at some basic level, there must be something to this idea that indeed genes in different categories tend to duplicate and delete at different rates. All right. So some nice follow-up work was done more recently by the group of Marco Cosentino Lagomarsino, where he showed that there is actually an intimate relationship between the power law distributions I found in my first biology paper, these gene family size distributions were power laws, and these scaling exponents. So remember, right, that if you looked at the distribution of gene family sizes, they made these power laws, and these power laws were less steep for large genomes. So in here, every line is a different genome, and in the color says how big this genome is. So the red genomes are the smallest, the yellow genomes are a bit bigger, the green genomes bigger, and the black genomes are the biggest. So you see there is this gene family size distribution gets less steep as you get to bigger genomes. And now they said, what happens if instead of looking at the distribution of gene family sizes, I'm only going to look at gene families that are transcription factors? So I throw away all other genes, and I just look at the gene family size of transcription factors. Then it's more noisy, but you also find roughly power laws. But these power laws have very different slopes from the power laws when you take all genes. And what you find is that this power law, beta C for this category, which is transcription factors in this case, is inversely proportional to the scaling exponent of this category. So that correlation is shown here in this plot. It's not a great correlation, but I think it's fair to say that indeed those categories that have a larger exponent alpha C tend to have also a larger 1 over beta C. All right, so that is to say, that for the transcription factor family, beta C is small, whereas for, let's say, translation, beta C will be much bigger. So it says there is some relationship between the distribution of gene family sizes and the scaling exponents of this, how you depend on genome size, right? And so any model that you're going to come up with for why there are these rules is going to also have to explain this relationship. And I should notice that to this day, I don't know yet of any, let's say, very convincing explanation for why scaling exponents are the numbers that they are. So there is a number of proposed explanations for why transcription factors are quadratic, right? The quadratic is a very sort of attractive thing to try and explain. But if you say, well, for transporters, the slope is 1.4, nobody has tried to explain that, okay? So this is still, I would say, to some extent, a largely open question. Yes? Yes, that is correct. Yes. So, yes, I will repeat the question. If you have a single domain, that domain may play a role in more than one biological process. Okay? And so in these scaling laws, this thing is counted twice. That's important actually because the first time, I showed this result to a very famous physicist, Murray Gell-Mann, and he said, that's a very strange polynomial you have there, right? Because it would say if the number of each gene scales as some strange exponent, but together they have to sum to just n, the total genome size, that's very strange, right? And so the way that works is because these things are overlapping. So that also says that somehow the amount of overlap must change as you change the genome size. No, I will get to that. Maybe, I don't know, but yeah. Yes, they have slope it's close to a half. Here's DNA repair. Okay, so you have to quadruple the genome to get twice as many DNA repair genes. Okay. Yeah. Yes.
Specification of functionality. Isn't it somewhat arbitrary? I mean, or,
Thank you for that question. Yes. So, how do we even come up with this functional classification? So I'll tell you how people came up with this. When people started sequencing genomes, they had people in their labs, bioinformaticians, that were doing the annotation, and that by hand would go through every single gene and write by hand, we think this is a transporter, we think this is a transcription factor. And of course, after a few years, when there were maybe a dozen genomes and people wanted to compare things, they realized people in different labs use different words to refer to things that we are thinking the same, right? So one writes regulator, the other writes transcription factor, but they're really corresponding to what we think is the same thing. So then these people got together and they said, we're going to form a consortium called the Gene Ontology Consortium, where we're going to have a controlled vocabulary with how we name things. And we're going to make a tree, basically a directed graph, it's really, where we define terms that can be specific cases, subcases of other terms and so on. So this is totally invented by humans, all right? And so indeed, there's some issue when you now want to extend this analysis. So I would argue that these kind of categories that are high-level categories that correspond to things that all genomes do to some extent, they are relatively well defined. I mean, it's always the case that if you say, oh, this transcription factor is actually a general factor that is every genome has and it doesn't do anything specific, do you want to put it in the same category as these very specific ones that regulate gene expression? Maybe yeah, maybe no. So at the edges of these definitions, there is always problems. But for these high-level functional categories, I think most people agree that they are fairly unambiguous. But once you drill down, and you say now, well, the transcription factor, it can be DNA dependent, but there can also be RNA dependent ones, and there can be activating ones, and there can be repressing ones, and you go and make it more fine-grained. Then it becomes incredibly human-oriented, and then it's kind of, that sort of makes it hard to do this analysis in a systematic way, because it kind of depends on the way that humans have carved up this functional space. But I think at the higher level, it is less ambiguous, even though it's still human-defined. But there is some sort of meaning to it that everybody... This is like asking about, you know, the yellow pages they give, you know, professions of people, right? I mean, yeah, it's human-defined, but it's not totally meaningless at some level of organization, right? So I think of this in a similar way. All right. This one. Okay. All right. So the broad message is this. So we found one type of power law distribution, namely the distribution of sizes of gene families. Right? How many genes, families of size 1, 2, 3, 4, 5? In a single genome. And we find that if, yes, beta is related to that. So beta is the slope, is the exponent of the power law of the gene family size distribution of all gene families in a given genome. And it depends on the size of the genome. So it's betas are smaller, or let's say less steep for big genomes. Yes, exactly. No, has nothing to do with this one. If you get from mixing slides from different talks, yes. Yes. You were talking about this. Actually, this one. This is the same beta. And S is the genome size, and they call that N, I think. Yeah. No, things that are evolutionarily related. So these are a set of genes that have sequence similarity where we can recognize these all belong to a family that came from a same common ancestor gene. Yes, exactly.
And things which are large, they are smaller in number.
Yes. Yes.
Larger.
Yes. Yes.
That's the
yes.
Beta then characterizes the genome.
Yes. And it depends on genome size. So for large genomes, the beta tends to be less steep than for small genomes. Here. Yes. Exactly. Right, so here, black are the biggest genomes, red are the smallest genomes. And now what you say, I'm going to do the same thing, but for a subset of all gene families. I'm only going to look at those gene families that encode for proteins that belong to category C. So for example, in this example for transcription factors, I'm only going to look at a subset of families that correspond to transcription factors, and I make another size distribution. And then I fit this again to a power law, and now I get a different slope, and I call that beta C. Now the observation is that beta divided by beta C, so the slope of this category relative to the slope of all families in the genome, is roughly equal to alpha C. And alpha C is a totally different thing. It says how the number changes with genome size. Okay, but they have some basic model of why they think that happens, and the model that I just showed you actually also makes such a prediction. So with the kind of model of this sort of biased reproduction and deletion of genes, you automatically get such things. Yes. 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 Let's say that's iteration 0 of the model. And now I know that there are certain problems with this model and so on. But that would take too long now to go. But yes, sort of the picture that I'm painting here is roughly that. Okay, so in the last time I have available, I now want to look at sort of the consequences of these scaling laws for the structure of gene regulatory networks. So, right, because the scaling law in the number of transcription factors tells you one piece of information about the regulatory networks. It gives an answer to the question, how many genes, how many transcription factors do you need to regulate a genome with G genes? And it says the number of transcription factors you need scales as G squared. Okay. I apologize again for the change in notation. This previously called N. Okay. G is just the total number of genes in the genome. And so far, everything I showed was for bacteria, but in eukaryotes, it is still true. So there is all kinds of troubles with defining what is a gene when you have a complex eukaryotic genome, and counting genes in different functional categories is a much more non-trivial exercise in higher eukaryotic genomes. But no matter how you count, the number of transcription factors is still increasing clearly faster than linear. And you can count it in one way and you get a slope as low as 1.4, and in another way you can get a slope as large as 1.8, but it's clearly going up linearly with genome size, even though it's probably less than quadratic. So one consequence of this is that this cannot go on forever, right? If I were to go to bigger and bigger and bigger and bigger and bigger genomes, and I extend this scaling law, so here this red line is a fit to how the number of transcription factors increases with the number of genomes, and the black genome size, the black line is just genome size. At some point it will cross. Okay, and so it predicts an upper bound on genome size. Now, it happens to be true that it seems that there is an upper bound on prokaryotic genome size. So I think the largest somebody has ever found is 12,000 genes or something like this. You can ask, does this thing correctly predict where this upper bound is? And if you do it naively in this way, you're off by about a factor of 3. So you predict that genomes can be 3 times bigger than they actually are observed to be. Now you can start sort of waving your hands and say, well, maybe the right way I should do it is that whenever I add one gene to the genome, I can add at most one transcription factor. Of course, that's not really true, right? Because I could add one gene, get rid of 5 transcription factors, and then add other things, right? But let's assume that I'm doing this incrementally, and every time I add one gene, I cannot add more than one transcription factor. Then that gives me a tighter bound, but it's still about a factor of close to 2 too large. But I would say that, okay, we're not off by 2 orders of magnitude. So I do believe that the fact that this sort of regulation is growing quadratically with genome size somehow puts a limit on how big you can grow. And we don't have it accurately enough that we can really predict the upper bound on genome size. But I do think that the upper bound of genome size is that sort of the administrating your genome runs at some point out of control, and you can't get bigger. Another consequence is that there are consequences for the shape of the topology of the gene regulatory network.
Sorry, Erik. So if you go back to the previous slide, what is the largest genome size we know, and how does it compare with the crossing point?
So for bacteria, it's about 12,000 genes today. I actually checked a couple of days ago what is the biggest one I can find. For long, it was sort of 10,000. Now it's sort of up to 12,000.
Okay, so
because this is all rescaled in terms of number of domains that have functional annotations. And so that's off from total number of genes by some constant. I'm hiding from you a lot of bioinformatic work that is very messy and that you have to do carefully to get right. But otherwise, this talk would be 3 weeks instead of 3 hours. Okay, yes. Yeah, no, that's a good question. I just think, okay, so the question was, okay, this is really stupid because I'm only looking at transcription factors. And I really should look at the scaling of all genes and should make some bound where sort of this thing breaks down when I look at all guys combined and say, I get some inconsistency that I can't satisfy. I agree, maybe the number is then, 1.5 times off, right? But we're within a factor of 2 already, and there's so much noisiness in this thing that I just don't think it's worth it to do this complicated model. It just says, it suggests that there might be a relationship between this existence of an upper bound and these sort of things that have to grow non-linearly, faster than linear with genome size. All right, so the last thing that I do, because I'm getting to almost half of my material, yes? Yes. Well, okay, so the fact that in eukaryotes you have genes cut up into exons and introns is one of the reasons why it makes it quite hard to estimate these numbers. And also, we know there is alternative starts, alternative polyadylation, alternative splicing. So now the question is, if one gene is spliced in 20 different times, should I count that as 20 or should I count it as 1? And depending on how you do that, you can get different, because it might be that transcription factors tend to have more alternative splice variants than non-transcription factors. And so if you count the alternative splicing variants, you get a different scaling law than if you don't count them, right? So it sort of enters into this thing, yes. I don't know. But maybe, okay, so, but I don't know. I mean, is it important? Because it would be really pity if I don't get to present this last part. Maybe after the talk. All right. Okay. Appropriate breaking point. All right. So we can think of the regulatory network of a genome as a directed graph, right? So in this simple cartoon, every gene is a ball. Genes that are not transcription factors are here in light blue, and genes that are transcription factors, I've given here other colors. And now the regulatory network is simply a graph where I draw an edge from the transcription factors to all the genes that they target. So this blue transcription factor targets this other transcription factor, this happened, right? And it targets these 4 genes here. All right. So now, oh no, one more here. Now I can count the total number of edges in this graph, total number of arrows, in 2 ways. I can go through all genes, and for each gene, look at how many inputs does it have. So this has 2 inputs, 2, 1, 2, 1, 1, 1, 1. And I calculate the average number of incoming edges per gene. If I multiply that by the total number of genes, I get the total number of edges. I can also go over the regulators and say, how many outputs does this regulator have? This one, this one, this one. So what's the average out degree for the regulators? And then multiply that by the total number of regulators. That's also equal to the total number of edges in this graph. But as I just showed you, the number of regulators is proportional to the total number of genes squared. So this R of G goes like R squared, G squared. If I plug that in, I see that this predicts that the ratio of the average in degree to the average out degree must grow linearly with genome size. Okay, so if you think of these regulatory networks as having some topological structure, it says this topological structure cannot be the same in small and large genomes, because the ratio of the average number of inputs per gene to the average number of outputs per regulator must grow linearly with genome size. And so there's 2 options, right? Either genes in larger genomes have just more regulatory inputs. That's one way of doing it. Yeah, you grow the number of inputs linearly with genome size. Or the regulators in large genomes have a smaller number of targets, right? You can decrease the number of outputs per regulator in large genomes, and you can also satisfy this. Or you can do some combination of these things, but the ratio has to be linear in genome size. So one of the things that one of my first students worked on was, what is it? And so the way we did it, and I will just sort of briefly skip over this, is we took sets of related genomes so that we're close enough that we could well align intergenic regions where these transcription factor binding sites are, and made alignments of intergenic regions, and in each of these genomes estimated by evolutionary analysis of the conservation of intergenic regions how many transcription factor binding sites are there per intergenic region, right? Because the idea is, if the number of inputs per gene is growing linearly with genome size, then if I go from a small genome with size 500 to a genome with size 10,000, the number of inputs per gene must go up 20-fold. I must put 20 times as many transcription factors. I should see that in that there is now more conservation near the start of the gene. Okay, so my student worked on this and really sweated over this for 3 years because he kept finding that there is absolutely no dependence on the amount of conservation upstream of genes on genome size. You don't see no signal where it looks like large genomes have more inputs per gene. And I was actually expecting this to be true. So I kept telling him, you're not doing it right. You haven't done the phylogeny right or the estimation right, redo it and so on. But eventually he convinced me. All right, so what we find is that there's really no dependence between genome size and the number of regulatory inputs per gene. So one way in which you can see that, what I'll show is you can just look at the sizes of the regions between neighboring genes in prokaryotes. So in a typical prokaryote genome, there is about 200 to 300 base pairs of intergenic region per gene. If you now ask, do these regions get bigger as you go to larger genomes? Do you get more intergenic DNA? You see there is no relationship at all. So these scatter plots show you number of genes, median length of the intergenic regions. There's absolutely no, and you would think, right, if you need to put 10 times as many binding sites, maybe you need some more room to put these binding sites. There's no evidence of that, right? And so we got convinced that the average number of regulatory inputs per gene in a genome with genes is not dependent on genome size. It's a constant. The consequence of that is that the number of outputs per regulator is scaling like 1 over genome size. So the picture is that in small genomes, so in each genome, there's the same number of inputs per gene. Each gene has on average 1 or 2 regulatory inputs. But in small genomes, there are very few transcription factors, and each of them target a large number of genes. Whereas in large genomes, there are many, many different transcription factors, and they each target a very small number of genes. So the picture, the cartoon here I drew, so here's a regulatory network. This is just a cartoon, right? But it's just to show you this idea. Here there are 10 genes total. 2 of them are regulators. There are a total of 10 regulatory interactions, 10 actions, 10 edges, 1 per input per gene. Every gene has 1 input here. And the average out degree of these regulators is 5. They have 5 edges coming out. If I now double the size of the genome, now I have 20 genes. I have to quadruple the number of regulators, right? Because it goes quadratically. So I have now 8 regulators, but the number of interactions is just doubling. There are 20 interactions, and still every gene has 1 input, like it had here. But now each regulator has half the out degree. Okay? So that's sort of the picture that we have of how regulatory networks change in their structure as you go from small to large genomes. All right, so the last thing I want to tell you, very briefly, is that this is not the case in eukaryotes. If you go in eukaryotes and you look at the size of intergenic regions as a function of genome size, you see that at some point it explodes. As you go to, when you go into animals, multicellular eukaryotes, the amount of intergenic DNA per gene becomes much, much bigger. And it's known, in fact, that in eukaryotes, one gene can be under the control of many, many different regulators, and that are from distal regulation of regulatory sites that sit up to hundreds of kb from the start of the gene. So there seems to be a very fundamental difference between the gene regulation in prokaryotes and eukaryotes. In prokaryotes have never invented the mechanisms to put one gene under the control of many regulatory inputs. So it can put a couple of binding sites near the start of the gene. It can put 1, 2, 3, 4, 5 maybe, and then it's game over. You can get no more complex regulation of this gene. In contrast, eukaryotes have developed in evolution mechanisms that allow to put one gene under the control of many different regulators. And so one question is, what is it about eukaryotes that they have that prokaryotes do not have that give them that ability? And my hypothesis is that this is chromatin structure. As you may know, in eukaryotes, the chromatin is wrapped around these nucleosomes that keeps most of the DNA inaccessible to transcription factors. And so in eukaryotes, what parts of the DNA are actually accessible for transcription factors to target is itself dynamically regulated. All right, so it's sort of like the genome that the transcription factors see is itself a function of the transcription factor expression. And I think it's that additional complexity in the regulation that allows you to put the gene under the control of regulatory elements that are far away. But I don't have time right now to tell you more about this. All right, so the second half of today's talk will be tomorrow then. Okay, and I'm going to talk about gene expression noise and how gene expression noise helps gene regulation to evolve. Okay, that's it. Thanks.
So didn't you just implicitly assume that cooperativity of transcription factors does not matter? Transcription factors may interact with each other and together give an input instead of binding separately?
Yes, that's true. But I don't, that's true.
Yeah.
So transcription factors can interact. I think the extent to which they do that in prokaryotes is not so clear.
Okay.
In eukaryotes, in eukaryotes, for sure, there are these interactions. I actually personally myself believe that a lot of interactions between transcription factors come from the fact that their binding sites sit under the same nucleosome, so they're competing for binding to the DNA with the same nucleosome. But certainly in prokaryotes, the extent to which this is important is not so obvious. But so how do you think it sort of affects the things I said?
No, so you said you ruled out that since the number of binding sites are not increasing,
Yeah,
input is probably not increasing to maintain.
probably constant, yeah.
Yeah. So if instead if it was not specific binding site, but binding on to another transcription factor which is already there and modifying its effect, that would be like a combined input of some sort, like just changing the input signal.
Yeah.
So something like that, instead of actually having the binding site on cis, but a trans binding sites on other proteins, on other.
Okay, so one thing we looked at, because one reviewer asked us, is, well, maybe in large genomes, you have many transcription factors, but maybe they fall into families that have very similar DNA binding domain and that actually compete for binding to the same sites. Right? So you see only one site, but it's actually a site for 4 different transcription factors because they all bind to the site. So we investigated whether it looks like in these big genomes, the binding sites might be binding sites for DNA binding domains that are very similar. And it doesn't look like that. So in fact, what we see, so the transcription factors in large genomes don't have more similar DNA binding domains. And if you go and look at the positions in these intergenic regions that show evidence of conservation, right? So you take your genome and you find all the places where you think there's conservation. And you now ask for the diversity of such, right? So you look at the sequences that occur there and look at how diverse do these sequences look. Then you see that this diversity is growing faster than linear with genome size, exactly as you would predict if the number of different transcription factors that are trying to bind there is going up superlinear. So this was actually, that part, when my student showed me that, that's when he finally convinced me that he was right. That it's...
Erik, maybe you said this and I missed this. So you assume dn by n and dnc by n are independent of time when you did that. Did that have a role in predicting the largest genome size? I missed the connection between the two.
No.
So even if this depended on time, you would still have a bound on the number of largest genome.
So the number of genes in the genome and in a particular category, they are changing in time. We know that. They're changing quite dramatically in time. What I'm saying is that this relationship is independent of time.
Okay.
So whatever DNs and DNCs you have, you can have growth, you can have shrinkage, but they're coupled. So how many more transcription factors you get or lose can be predicted from the change in the other way around. How many transcription factors you get added is a function of the total number of genes added.
So it's a very interesting point you make for the difference between prokaryotes and eukaryotes, right? So it's one gene under the control of many regulators.
Yeah.
And it reminds me of this ecological scaffolding that Professor, maybe you weren't there, I think.
No.
But right.
I'll try to answer nonetheless.
Right. So it's like a collection of conditions that make your particles together, right? So in this case, there will be your gene. So what do you say could give rise to such an interaction then?
How do you get a gene under the control of many regulators? Okay, so I'll give you my physicist's spherical cow picture of gene regulation in higher eukaryotes. So in higher eukaryotes, everything is turned off by nucleosomes covering the DNA and closing it up. All right? Now there are places where there are combinations of binding sites for transcription factors that happen to be co-expressed in this condition. And then these transcription factors together can outcompete this nucleosome that is sitting there covering the DNA. Right? So most places of the DNA do not have combinations of binding sites for the transcription factors that are co-expressed in this tissue type. But the small fraction of places where there is such a thing, there the nucleosomes will be ejected and the DNA will be accessible. And it will be accessible for the RNA polymerase to come and bind and start transcribing. And so if there is a transcript near such a place that has been opened up by the transcription factors, you will get that transcript and you will get gene expression. Now, in eukaryotes, there are also places where there is no transcript. So the polymerase will come and it will try to transcribe, but it basically will keep falling off and keep being stalled because there's no transcript to go. However, if such a polymerase that has been recruited to such a place gets close to another open place where there is a transcript, the polymerase can be transferred from this open place to the other open place, and then you will get transcription. And this is what is called distal regulation. So one of these open places is called an enhancer or a cis regulatory module. And it will just open up, and it's just like a promoter, it's just that there is no transcript. And so the polymerase will come, it will get all ready to transcribe, it will even try to transcribe. You can see that if you sequence deep enough, you see these little reads coming from these enhancer regions, but it cannot stably transcribe from there. And so it has to wait until, flopping about in the cell, it hits another open region where there is a transcript, and then the polymerase can be transferred and transcribe that gene. And in this way, you can have one gene not being regulated by the binding sites near it, but also other distal regions that get opened up can deposit polymerases here. Okay, so it's way more complicated than that, but that's my sort of zeroth order model of how this works in higher eukaryotes. Does this sort of answer your question?